Hello everyone, welcome to this first lecture on the neuroanatomy of the spinal cord. To contextualize the topic a little bit better, we will commence by talking about some basic neuroembryological facts. Next, I'll elaborate upon the gross anatomical features of the spinal cord. Then we will briefly take up the descending motor and the ascending sensory spinal pathways one by one. The detailed course of these neural pathways and their important functions will be discussed in some subsequent lectures on the spinal cord. The knowledge acquired from this series of lectures on the spinal cord will help you to apply this basic neuroanatomical information to localize neurological lesions in the nervous system. This will also prepare you nicely for your neurology clinical skill stations and your neurology placements in the hospital. Although the details of the embryology of the nervous system will be discussed in a separate lecture on neuroembryology, however, I think it will be useful to discuss some basic neuroembryological facts prior to discussing the anatomy of the spinal cord. So let's start with this illustration, and how about if we orientate ourselves first? What you're looking over here is a cartoonic illustration of a coronal side-to-side section of an elongated human embryo. If the arrow in blue over here shows the longitudinal or the anteroposterior axis of the embryonic body, then this means that the head end or the rostral end of the embryo will be towards us over here, kind of popping out of the screen, while the posterior end is going to be away from us. The embryo has its back onto the dorsal side over here. This is the dorsal aspect of the embryonic body, and the ventral aspect of the embryonic body is going to be over here. This would be the right side, and this would be the left side of the embryonic body. Now, we know from our knowledge of embryology that the human body is formed of three germinal layers. You can see the three germinal layers over here. The outermost layer here is the surface ectoderm. It is followed by the mesoderm, which is the intermediate layer, kind of color-coded in brown over here, which is then followed by the endoderm, which is the innermost layer. The endoderm is going to line the gastrointestinal tract. Therefore, this would be the anterior or the ventral aspect of the human embryo, while this would be, obviously, as we discussed, would be the dorsal aspect or the back of the human embryo. By the beginning of the third week of development, the surface ectoderm then thickens out to form what is known as the neural plate. You can see the thickening of the surface ectoderm over here. The lateral edges of the neural plate will elevate to form what are known as the neural folds, creating a neural groove in the midline over here. With further development, the neural folds will continue to elevate, and you can see the neural folds elevating over here. They will approach each other in the midline and finally fuse together to form the neural tube. The fusion commences in the cervical region but then proceeds both in the rostral as well as in the caudal directions to form a cylindrical neural tube. The tube eventually buds off from the surface ectoderm, and you can see that over here. The surface ectoderm is at the top, and the neural tube has bud off from the surface ectoderm and has come to lie at a slightly deeper level, at the level where the mesoderm is situated. This neural tube is eventually going to form the central nervous system, including the brain and the spinal cord. Now, some of the surface ectodermal cells, they thicken out to form what is known as the neural crest. So you can see the neural crest cells over here, color coded in green. Like the neural tube, the neural crest cells, they are also going to bud off from the surface ectoderm, and they will come to lie at a deeper level just above the neural tube and underneath the surface ectoderm. These neural crests are then going to differentiate into specific parts of the nervous system, such as the dorsal root ganglion and many more. Now I'm going to talk about the neural crest cells and the neural tube in a little bit more detail. So here you can see a cut section of the neural tube, color coded in red over here, along with the neural crest cells shown in green over here. So what happens is that the cells of the neural tube or the central canal, they would start proliferating to create three distinct zones of cells around them. The innermost layer of cells, which are forming the lining of the central canal, they are referred to as the ventricular zone of cells. The outer layers of cells, shown in blue, they constitute the mantle zone of cells, while the outermost layer of cells, shown in purple or kind of gray over here, they constitute the mantle zone of cells. The central nervous system, which comprises of brain and the spinal cord, that is going to form from these three different zones of cells. This is an illustration of the intermediate stage of development of the spinal cord, while this represents an adult stage of the development of the spinal cord. The central canal gives rise to the ventricular system of the brain and the spinal cord, such as the lateral ventricle, the third ventricle, and the fourth ventricle. Specifically in case of spinal cord, this will form the spinal canal. Cells in the ventricular zone form the ependymal lining of the spinal cord and the entire ventricular system. Next, the cells in the mantle zone, they form two types of plates called as the basal plate, situated ventrally, and the alar plate, situated dorsally. This is an illustration of the horizontal cut section of an adult stage spinal cord. The central butterfly-shaped area over here, this is the gray matter. The two ventral horns are formed from the basal plates from over here, and these are motor in nature, while the two dorsal horns over here, these are formed from the alar plates at the back, and these are sensory in nature. A motor root emerges from the ventral gray horn, which is a neural tube derivative, while a sensory root, which enters into the dorsal horn, is a neural crest derivative. Both the roots, they then combine together to form what is known as the spinal nerve. Lastly, the outermost zone of cells, which are the cells present inside the marginal zone, they develop into the white matter. You can see in the transverse section of an adult stage spinal cord over here that the part of the white matter which is in between the two anterior horns, that is known as the anterior white column. The white matter in between the two posterior horns over here, that is known as the posterior or the dorsal white column, while the white matter which is lying in between the anterior and the posterior horn, that is the lateral white column. Now let's see where the brain and the spinal cord are situated. For that, let's take the help of a prosection. What you can see over here is a prosection which shows the posterior view of the central nervous system, including the brain at the top over here and the spinal cord down below here. See, the cranial vault has been removed from the back, and you can see the meninges which are wrapping over the brain. You can see the back of the brain over here. So this is the occipital lobe. We can see the right occipital lobe and the left occipital lobe over here being separated by a dural partition which is known as the falx cerebrum. Then we can see a horizontal partition of the dura mater here, which is called the tentorium cerebellum, and that is going to separate the occipital lobes at the top from the cerebellum down below. Now, the cerebellum, which was originally residing inside the posterior cranial fossa, has been removed. Once we take out the cerebellum, we can see the back of the brain stem. The lower part of the brain stem, the lowermost part of the brain stem, is the medulla oblongata, and you can see the medulla oblongata passing through a big hole in the floor of the cranial cavity. The hole is known as the foramen magnum, and once the medulla oblongata passes through it, it enters into the vertebral canal, where it is now the spinal cord, and you can see the spinal cord being housed nicely in the vertebral column all along its length over here. Now let's talk about the gross anatomical features of the spinal cord. Let's resort to this prosection over here. Here you can see a prosection showing a mid-sagittal view of the vertebral column. The vertebral bodies can be seen in the front over here with the intervertebral discs in the middle. So this would be the anterior side of the vertebral column of the vertebrae, and we can see the spinous processes at the back, so that will be the posterior side. The spinal cord can be seen nicely resting inside the vertebral canal. The image on the right over here is a cartoonic replica of the prosectional image which we just saw over here, where you can once again see the spinal cord resting inside the vertebral canal. We can see the bodies of the vertebrae in the front, and we can see the spinous processes at the back over here. We can use this illustration to highlight some of the important gross anatomical features of the spinal cord. For instance, let's examine the extent of the spinal cord. In the case of adults, it can in case of adults, the spinal cord extends from the foramen magnum to the inferior border of the first lumbar vertebrae, the L1 vertebrae, which we can see over here. In case of newborns, it can extend as far down as the L3 vertebrae. You can probably appreciate over here that although the spinal cord ends at the L1 vertebral level, however, the meningeal wrappings which cover over the spinal cord and are marked over here by this black line, they extend down to as far down below as the second sacral vertebral level at the S2 level, which technically implies that the vertebral canal below the L1 vertebrae and above the S2 vertebrae will contain subarachnoid space, which contains the cerebrospinal fluid inside it. However, there would be no spinal cord in that region, thus making it a safe zone to perform a lumbar puncture for diagnostic or for therapeutic reasons. As far as the shape of the spinal cord is concerned, the spinal cord has an elongated structure. However, you can probably appreciate that the diameter of the cord is not constant throughout its entire length. The cord is dilated in the cervical region and in the lumbosacral region shown over here. These are known as the cervical enlargement and the lumbosacral enlargements of the spinal cord. The cervical enlargement extends from the C5 to T1 segments of the spinal cord, whereas the lumbosacral enlargement extends from the L1 to S2 segments of the spinal cord. And the cord is unusually enlarged in these regions, in the region where the cervical enlargement and lumbosacral enlargements are present, due to the extra number of neurons to cater for the innervation of extra muscle tissue inside the upper limbs and the lower limbs respectively. Lastly, before the cord terminates, it tapers off to form this conical ending which is known as the conus medullaris. This is an important region of the cord which contains fecal and urinary control centers of our body. A lesion in this region could potentially lead to urinary and fecal incontinence.